condition is to be health condition, healthy condition, that is good health, good mind, good health, good condition, these are to be occur. When health is to be good, mind is to be good, our condition these are to be good. When animal become pathogenic, then their mind is also changed, these are to be disturbed. That's why we have told that is sound health, sound mind, similar fashion, control measures of diseases, these are to be essential to understand which are the causative reasons, which are the causative points behind these pathogens, pathogenicity. For that, this control measure, these are to be important, these are to be, we are studying here. See, this is the figure or the, in this plate, in every insect, we have seen, we can observe different fishes. Which are these fishes? These are to be observed, these are to be seen, these are to be occur. First, this is the gas bubble disease, which is to be occur. Fish, these are to be caused by the disease, these are to be occur due to the intrinsic factor, due to the nutritional diseases, that is the gas bubble. In second figure, this is the anchor worm. This is the anchor worm. This anchor worm, these are to be seen, these are to be observed on the goldfish body. This is another parasite, another pathogen. In third fish, that is black muley observed. This is the aquarium fish. On this black muley body, pinpointed white spot, these are to be appear, white spot, these are to be occur. That is due to the protozoan parasite, due to the each spot or due to the Ichthyophilus multiplicis, this black muley, these are to be infected and cause the disease. Second, that is a tail dot, or see, this is the tail dot, that is caudal fin, these are to be brittle, these are to be cut down, bacterial disease. Again in this fish, that is cotton mouth disease, these are to be occur. Cotton mouth disease, these are to be occur, that is these are to be found, these are to be seen in this fish, that is mouth, these are to be eroded, mouth, these are to be infected. In next figure, in this figure again see, complete caudal fin, these are to be brittle, and when caudal fin, these are to be brittle, these are to be damaged, cut down, then movement or swimming of fish, these are to be affected, these are to be infected. All these figures, these are to be indicate that all these insect figure, these are to be clearly shows that fishes that may be caused, fishes that may be infected due to the infectious diseases as parasites, infectious diseases as protozoan, fungi, bacteria, virus, protozoans, crustaceans or helminth. Whereas non-infectious diseases, that is food due to the deficiency of food, due to the deficiency of vitamins, deficiency of protein, fats, carbohydrate, fishes, these are to be infected. Infectious, non-infectious diseases, which is to be seen in this figure, that is gas bubble. All these show that fishes, these are to be severely infected or affected due to the eccentric parasite, due to the ectoparasite as well as the endoparasite. To control all these parasite, control measures, these are to be essential, control measures, these are to be most important. That is, prevention is better than cure. Prevention is better than cure. Similar fashion, these symptoms, these diseases, these are to be controlled, these are to be minimized, these are to be lowering. We can perform, we can control these diseases, these pathogenic activity, and for that purpose, control measures are best way to know, to understand the expert, to know fish culturists, fish farmers, prawn culturists, prawn farmers, crab culturists or the crab farmers as well as the molluscan animals They can be cultured, that may be Mytilus, that may be sepia, that may be loligo, chang, squids, octopus, all these animals where these are to be cultured, these are to be grown, that culture is, it have well-known knowledge about the pathogenicity, about the disease, then and then control measures we can perform. And when control measures these are to be done in a advanced time, in a proper time, then loss of crop is to be avoided, loss of fish production is to be avoided, and for that purpose control measures of fishes, control measures of pathogens or the fish diseases is the important aspect, is the important point to understand, to study. For that purpose this article, these are to be important, these are to be most useful. Control measures of fish diseases. It is correctly said that an ounce of prevention is worth a pound of cure. What is the phrase? A phrase is to be said everywhere. An ounce of prevention is worth a pound of cure. Minimum infection these are to be occur. For that purpose maximum investment, maximum money they can waste, they can spend to avoid this condition, that is prevention is better, prevention is most important. It is correctly said that an ounce of the prevention is worth a pound of cure. A sound management of the fish farm may lead to the prevention of a large number of fish diseases. A sound management, which is to be important, management of diseases, management of pathogens or pathogenicity, these are to be most important for the prevention of fish pathogens, fish parasite. It is to be well known for the fish farmers, fish culturists. That's why control measures of fish diseases we can understand, we can better known. Hence these are important, these are most useful. Following some measures were taken, considered seriously, prevent most of the fish diseases. Following measures were taken when considered seriously, when fishes these are to be seriously infected, seriously affected, then some measure we can taken, some measure we can take, and we can control, we can cure these pathogens, these fish diseases. Which are these control measures? The first control measure, disease should be detected early and proper. Disease should be detected early and proper treatment initiated immediately. When we have detect early in period, early in time, pathogens, pathogenicity and proper treatment, these are to be initiated immediately. As fish farmers, they have known fishes these are to be infected by bacteria, fishes these are to be infected by virus or fungi. Immediately treatment should be started, treatment should be immediately initiated, because it will take a short time for a disease to infect the entire water bodies or ponds. Suppose we do not take care, we do not alert of pathogens or this parasite, then within short time, within quick time, these pathogens, these parasites, these are to be spread all over the water bodies, and complete crop these are to be infected, complete crop these are to be affected, and then it is to be severe, it is to be more difficult to control such condition. For that purpose, that is early detection of the pathogens or disease, these are to be essential. And as we have known about the disease or pathogens, immediately, that is treatment should be initiated, start, because it is to be within short time, within short period, these are to be spread. Hence immediate treatment and proper treatment these are to be given, proper treatment these are to be start initiated to control the pathogens, to control the fish diseases. Next, control measures when the pond or water body has infected before harvesting. When the ponds or water body was infected before harvesting, the disinfection should be done with potassium permanganate. We have understood that is the ponds or water bodies which is to be infected. Before harvesting, fishes was infected, fishes was affected by different pathogens, different parasites, before harvesting, the disinfection should be done with potassium permanganate solution or potassium permanganate solution. These are to be important, these are to be most used. Potassium permanganate, here is the potassium permanganate. Potassium permanganate solution, and this potassium permanganate is to be important to cure the pathogens or the diseases. Next control measure, the bottom of pond should be thoroughly plowing and drying is done till the bottom of the pond should be thoroughly plowing and drying is done till the cracking of soil followed by the liming. As pond or as water bodies, these are to be infected by parasite pathogens. Complete water bodies, these are to be plowing, and plowing is to be done. After plowing, the water or that tank, that pond should be drying is to be done. How much time these are to be drying? Till cracking of soil. When soil, these are to be cracked, and due to cracking of soil, that is oxygen, these are to be mixed into the soil, and these pathogens, these parasites, these are to be killed by the heat of sun, as well as they can killed by the lime. Followed by this treatment, followed by this cracking of soil, wood, ooze or mud, these are to be occur. Liming, these are to be sprayed on the mud or bottom. When liming, these are to be sprayed on the mud or bottom, those parasites, those pathogens, these are to be grown. These mud or these ooze, these pathogens, they can dead, they can paralyze, they can completely controlled. When the bottom of the pond should be thoroughly plowing and drying is done till the cracking of soil followed by the liming. As cracking, these are to be done, liming should be occur. Liming should be, that is lime, these are to be applied, and that lime is to be near about 150 kg to 200 kg per hectare area. These are to be applied, these are to be sprayed. How much lime these are to be applied? Lime, these are to be applied 150 kg to 200 kg per hectare area. Lime, these are to be sprayed. Lime, these are to be completely used by the cracking on the mud or ooze or bottom of the water body. Next control measures, quality of water and food should be thoroughly checked. Again important, when we have cultured fishes, when we have cultured aquatic animal, that may be fish or that may be non-fish organisms such as mollusks, arthropods, prawns or etc. When we have cultured these animals, these organisms, then quality of water should be regularly checked by regular expert person. Expertization, those have expert knowledge, about the water quality. To this expert scientist, expert person, that expert person, these are to be continuously checked the quality of water, and those food which is to be supplied, as well as those food which is to be provided in that water body, in that tank, that food, these are to be checked regularly. That food may be supplementary food, that food may be natural food which is to be checked by expertization, by expert person. It is to be controlled, it is to be cure the fish pathogens or diseases. Second control measure, the predatory and debris should be carefully removed. Those predators, that may be those predator animals, those predatory animals, that may be fish, that may be other animals, that may be frogs, or that may be reptilian or any different predatory animals, that predators and debris should be clearly removed. Debris, waste part, fallen part. When these predators, these are to be removed, fishes, these are to be freely live, and those pathogens, those parasites, these are to be spread by these predators. They can avoid, they can not spread. And second, debris, these are to be possesses dead and decaying waste materials. Dead and decaying waste material, and when these dead and decaying waste material, these are to be stay for long period, stay for long time, it can pollute the water. Foul odor of water, these are to be occur, and when water pollute, foul odor, these are to be occur. Ultimately, number of fungi, number of bacteria, number of microorganisms, they can grow, on such debris. That's why these debris should be carefully removed time to time, regular interval, which is to be important to control to cure the fish pathogens or the diseases. Next control measure, the behavior of fishes should be constantly observed. Those are the culturists, those are the scientists, those are the workers which is to be working on the fish farm, on the fish engineering farm. These persons, they have knowledge about the fish and their behavior, fish and their survival condition. The behavior of fishes should be constantly observed. Suppose any fish these are to be behave abnormal to their normal behavioral condition, then at that time or such time, such period, the fishes they can check, fishes they can watch, and when abnormal behavior occur in the water bodies in tank, then we can remove such fishes and examine, that is examination these are to be done, diagnosis these are to be done, whether it is to be abnormal behavior, why these are to be performed. Then and then we can constantly observing and noting the behavior of or behavioral changes. We have known the pathogens or the parasite. This is another important control measures to control these diseases or these pathogens. Next control measures, a regular watch for the trouble and diseased fish is the last step for the healthy growth of the fish. What is the control measures? A regular watch for the trouble and diseased fish. Those fishes these are to be trouble, those fishes these are to be stressed. Those fishes these are to be trouble and diseased. Such a regular watch these are to be done. Regular watching these are to be important, which is to be diseased fish is the last step for the healthy growth of the fish. Those fishes these are to be healthy grown. Those fishes these are to be grow healthy. For that we can watch trouble and see the fish carefully whether these are to be infected or it is to be healthy, which is to be free from parasite or the pathogen. Next control measures, any change in normal behavior. Any change in normal behavior may be sign of disease. May be sign of disease. Any change in normal behavior. Normal sign of fish, normal sign of organism, these are to be changed. And when normal size of organism, normal behavior of organism, these are to be changed. It is the signal, it is the sign for the disease, it is the sign for the trace. And when a trace these are to be increased, disease these are to be increased, then fish show the repeated signaling, repeated such behavioral changes. For example, rubbing the body. When fish these are rub their bodies to the dike, to the bottom, to the any obstacle which is to present in water, it indicates that fish these are to be infected by protozoan parasite, fish these are to be infected by worm parasite, fish these are to be infected by helminth parasite or the other different parasite. When they can rub body, some parasite pathogen they can fall down. We have seen already in different fish diseases. Second, gasping of breath. When dissolved oxygen level these are to be minimum, dissolved oxygen level these are to be low, and fish these are to be jumping outside the water to take the atmospheric air, to take the atmospheric gas, that is gasping for water. Fish they can jump, fish they can jump. These are to be a signal, these are the symptoms for pathogenicity or the disease condition. Or suppose food these are to be supplied, supplementary food these are to be provided, and fishes they can do not take food, do not consume these supplementary food. Again they can refuse to eat the food, then it is to be a signal, it is the chance, because when fish they refuse the food, when their health, when their ill condition these are to be occur, normal behavior these are to be changed, and fish intrinsically these are to be behave abnormally and they refuse their food eating condition, it is a symbol, it is a symptom. That's why normal behavior these are to be changed, these are to be watched by expertise person. Next control measure, the parasite usually become attached on the body. Parasite these are to be attached, infected on the body. Parasite these are to be infected on the gills. parasite, these are to be infected on the fins, parasite infected on the skin, infected on the mouth or any different part, these parasites, these are to be infected, these are to be affected. Fishes can be easily removed and killed by use of certain chemicals. Such infected fishes, such affected fishes, that part that is either tail portion, these are to be brittle, either fin areas, these are to be brittle, either mouth, these are to be brittle, or gills, these are to be choked up and cut down. At that time, parasite usually becomes attached on the body. Fins, gills or fishes can be easily removed and killed by the use of certain chemicals. Those infected fishes, these are to be occur. These infected fishes, these are to be removed, and with the help of certain chemicals, with the help of various types of chemicals, we can control, we can kill all these parasites. It is the another important control measures. Then next, a common method involves the use of about 3% salt solution to control parasite pathogens. To control parasite pathogens, a common method, these are to be involved, and that common method is nothing but the use of 3% salt solution, sodium chloride, that is common salt which can use for eating. This common salt use 3% to kill, to remove the pathogens, parasite. It is the good control measure, good method. Next method, other chemicals can be used. Other chemicals, not only salt, these are to be used, not only salt, these are to be utilized for control of pathogens, parasite, but other chemicals such as copper sulfate, such as potassium permanganate, such as acetic acid, formalin, etc. Means copper sulfate, potassium permanganate, acetic acid, formalin, all these chemicals, these are to be involved, these are to be used to control, to minimize the pathogens or parasite in a proper range and proper level, because again when it is to be excess, harmful for the fish or the aquatic animal. For that purpose, these chemicals, these are to be used whenever we can need, whenever we can emergency to apply these chemicals. Such chemicals, these are to be spray or these are to be mixed into water, and we can control the parasite pathogens. Next, many external parasites can be killed by using dilute solution of acetic acid. Many external parasites, those parasites, these are to be ectoparasite, attack on the fish body, attack on the fishes. That may be worms, that may be louse, that may be lernia, or that may be helminth. Such many external parasites can be killed by using a dilute solution of acetic acid. When these infected fishes, these are to be dipped into acetic acid solution, then these parasites, these pathogens, they can paralyze and they can fall down, remove from the fish body. With this control measure, we have control, we have minimized the pathogens or the fish parasites. Next, control measures, mechanical removal of the parasite can be done. Mechanical removal of parasite, it is to be done, but it is to be risky and time consuming again. It is to be risky and time consuming. Why it is to be risky? How much time we can keep fish out of water? One question. And second question, how much fishes we can remove the fish? Mechanical removal is to be best way. Mechanical removal is the good method to control, to minimize, to lower the fish pathogens or the fish diseases. But how much time? Individual fish, these are to be caught, these are to be captured. Then parasite, pathogen, these are to be removed by forceps, by hands, or how much time fish, these are to be out of water, these are to be live, survive good, healthy condition. For that purpose, it is to be difficult, but it is to be another way, good method, mechanical removal of parasite can be done, can be performed for the control measures. These are the some control measures to control parasite, to control the, that is nutritious diseases or the infectious diseases. These infectious diseases, parasites, and these diseases can be controlled. See in this figure, we can observe some fishes, these are to be infected by parasite, that is leech. Some fishes, these are to be infected. These are the parasite, these are the pathogens. These are the parasite, or these are pathogens, these are to be infected by the worms, by the eccentric, parasite, that is various. And these are the, that is diseases, these are to be occur, gas bubble disease, or that is these are to be infected. All these are the another disease. All it indicates that these diseases, these can be controlled, cure with above different control measure they can adapt, we can use, and then we can control, we can cure the pathogens or the diseases. With this we have we have seen the control measures of fish diseases or the fish pathology, that may be parasitic or that may be non-parasitic. Hope you have understood and well studied up to here. Thank you.